My name is DéJenaé See. I'm a transfer student and a graduating senior at UC Berkeley. My major is chemical biology and I'm minoring in bioengineering. I am also an undergraduate research assistant in the lab of Dr. Caroline Williams in the integrative biology department. So my SURF fellowship project was focused on characterizing proteins in this tardigrade species Hypsibius exemplaris. Tardigrades are commonly called water bears. They're these microorganisms that only grow about a millimeter in length, and they are found in all sorts of environments, especially in aquatic environments and around mossy areas. They're mostly known for being able to withstand a lot of environmental stressors. So you might have heard of tardigrades being used as experimental subjects in space because they're able to handle like low pressure environments, lack of oxygen, depending on the species. And our species is especially good at being able to survive for long periods below freezing. So the tardigrade that we were looking at, while its proteome had been sequenced, not much had been known about the different types of proteins, and we suspected that ice binding proteins might be the reason why these tardigrades do so well below freezing temperatures. The challenge with this was that these proteins, their amino acid sequences don't really have any homologs. So in other words, the proteins that we're interested in have no similar proteins where the structure is already known. So we had to use a platform that would basically read the amino acid sequence and from that alone give us the protein structure, predictive protein structure. So that was my job this summer, was building on our lab's prior work doing proteomics experiments to try to characterize candidate ice binding proteins using platforms that were based on this very popular protein modeling software called Rosetta. And using that, we've been able to isolate a handful of proteins that are probably responsible for, or possibly rather responsible for, being able to, for tardigrades being able to survive sub-freezing temperatures. We are now moving into the stage of trying to clone these candidates and sending these off to a partner researcher in Canada that will help run something called thermal hysteresis experiments that will tell us a little bit more about how these proteins actually work. So that is the ongoing research for this semester. So I first got involved in the Williams lab actually as a work-study student in animal husbandry and worked my way up to being a research assistant, and the SURF program gave me the opportunity to continue through the summer with a paid summer project. And this work really matters to me because it's experience with understanding the relationship between protein structure and function and how computational biology plays into that, which is really relevant to my larger career goals because I'm interested in the potential protein engineering as it can apply in biomedical research. So this was a great opportunity to really get acquainted with that and get my foot in the door. So that's it, and thank you for listening. I hope you all stay healthy and stay safe, and go Bears!